to record, share, PowerPoint. All right, and let's go here. Okay, so can you all see my camera or the PowerPoint right now?
Yes.
I'm sorry, which one? Can we see PowerPoint or? Fantastic, great. Okay, so you all are connected through the telephone. So if you have any questions along the way, just straight up interrupt me. That's the only way that I'm gonna know you have any questions, because I can't watch everyone's video footage as I'm giving the lecture. So again, just you have anything to say, you can either wait till the end, but I would prefer that you just kind of interrupt me and just say something, whatever you need to do, okay? All right, y'all, ready? Let's do it. Okay, so we are learning some basic chemistry today. I don't know what you all have been exposed to in the past. If you've taken chemistry since high school, we're going to go through basic organic chemistry today. So why are we going to learn this? This is from the beginning and end of your chapter. So it's going to allow us to understand, as they put it, the daily lives of the microbial workforce. It's going to help us understand how microbes survive and how we interact with them. And then at the end of the chapter, they mention how this is kind of complicated material. It doesn't seem like it's directly applicable to microbiology, but I promise, learning about peptide bonds, learning about hydrolysis reactions, this is going to come in useful for later chapters, promise. All right, so let's just get started. By the way, just to give you a heads up, today's lecture is going to take maybe 40 minutes. So
I have a quick question.
Yes, please.
Sorry to interrupt you.
No.
The chapter 2 note says this file is part of an unpublished module and is not available yet.
Okay, great. Give me 1 second. Let me go back in.
I don't want, okay, thank you.
No, really, it's no problem.
Okay.
So let's go exit full screen, back here. I'm still getting used to Canvas. So let's go, okay. So modules. Okay, so I just clicked the button. It should be available now. Will you go ahead and check for me, please?
Okay, I can see it now.
Awesome. Thank you for letting me know. I appreciate that.
Thank you.
Also, great example of how to just step right on in.
Thanks.
Okay, so I just want to make sure you all can see PowerPoint again, yeah?
Yes.
Okay, great. Thank you.
No.
All right, let's get started on chapter 2.1. So organisms are comprised of matter. We exist in the material world, right? And all matter is comprised of atoms. Atom is the fundamental building block of matter. You go below an atom, and we're going to be talking about particles. You've probably heard these be called subatomic particles. So things like a proton, a neutron, and an electron are all particles that comprise atoms. In the nucleus of an atom, we're going to have protons and neutrons. They're going to be about the same size, but a proton carries a charge. It's going to carry a positive charge. A neutron is neutral. Electrons are going to balance out the positive charge of a proton. They carry a negative charge. So you'll often see it as an E with a negative sign.
Hey, Nick.
Yes?
We can't see the PowerPoint in full screen.
You cannot?
Nope.
No,
okay, hang
we
on.
see your back screen on the wall.
Yeah, that's all we can see. We can see everybody else.
How about that? Is that better?
No.
No? Okay.
I don't think you're sharing your screen. I think that's what it is.
Okay, cool. Share and PowerPoint, and then go slideshow. Okay, now how are we?
That did
Much
it.
better, thank you.
Perfect, thank you all. All right, so atoms. We have protons, neutrons, electrons. The protons and neutrons are in the nucleus, and the electrons are going to be in these outer shells, is what we're going to call them. As we go further away, we have higher energy in the shells. You'll notice that the shells have different numbers of electrons. So for example, the first shell around the nucleus can only allow 2 electrons to fit within it. If there are 2 electrons, we're going to say it's full. And then we're going to start building outside in our second shell. Once that becomes full, we'll move outside further. This isn't a chemistry class, so I don't need you to memorize the number of electrons or the shapes of the electron fields. But you should know that if an outer shell is full, we're going to go ahead and say that it is unreactive. If it is not full, it is reactive. It is unstable. So an atom, by definition, is based on the amount of protons in the nucleus. Hydrogen has 1 proton. Let's see, we have our periodic table right here. Helium has 2 protons, etc. So if you have more or less protons, you're a different element at the basic level. But if we have a different number of neutrons, we're going to call that an isotope. And if we have more or less electrons, we're going to call that an ion. Both of which are going to cause a different charge in the overall atom. And so we just mentioned that if an atom has an unfilled outer electron shell, whichever shell that is, it is unstable and more reactive. If it is filled, it is more stable and less reactive. So here, this figure shows magnesium losing 2 electrons. So if electrons carry a negative charge, you lose 2 of them. That means you now have 2 more positive charge difference than you did before. 2 more positive charge difference. That's worded poorly. You have more of a positive charge because you lost some of your negative charge. So you're no longer neutral, you're carrying positive. Okay, and then I really like this figure. I definitely recommend you all study this for your quiz coming up this week. So let's just take a look at this. An atom has a nucleus and electrons. Protons carry a positive charge, more or less, different element. Neutrons, more or less, isotope differences. And if we add or remove electrons, we have different ions. If you have a positive ion, we're going to call that a cation. If you have a negative ion, we're going to call that an anion. One more thing to note is if we take an atom and attach it to another atom, we're going to call that a molecule. It can be the same or a different atom. So can you all think of an example of a molecule that has 2 of the same atoms? So water is going to be a molecule that is comprised of different atoms. So we have 1 oxygen atom and 2 hydrogen atoms. But something like O2, that is molecular oxygen. It's 2 oxygen atoms bound together. And that's a gas. So they can be the same or different atoms to form molecules. But the definition of a molecule is 2 or more atoms attached to one another. We're going to talk about a couple different types of bonds, primarily ionic and covalent bonds. In an ionic bond, we're going to form through donation and receiving of an electron. So one atom will give another atom an electron. That's going to give the atom that donates the electron, so it loses some negative charge, it becomes positive. The atom that receives an electron picks up more of a negative charge. So now we have an attraction between positive and negative, just like two magnets. And so you can see here in this red band, that shows the electrostatic attraction between the positive and the negative atoms. And again, keep in mind this happens in the outermost shell of electrons. So that's an ionic bond, a covalent bond. So think co meaning together, valent. These electrons, we call them valence electrons. So electrons in the outer shell. If you are covalent, that means that you are sharing electrons. You're not donating and receiving, you are sharing them. And here we have an example, methane. So this is one central carbon bound to 4 protons. So here's the simplified stick structure. And carbon is very reactive because it has 4 valence electrons. So it can form up to 4 binding partners. So we're going to see throughout today, carbon and hydrogen interact very, very often. So these covalent bonds, they can form either polar or non-polar molecules. You want to take a stab at it? Explain what polar and non-polar means? So that's one way to think about it. Water is a polar molecule, so it will dissolve other polar molecules. Things that are non-polar will not dissolve in water. But how about the word polar, what that means?
The large electronegativity difference.
I'm sorry, what did you say?
I said a large electronegativity difference on each end of the molecule.
Right, there's a difference between electronegativity on one end of the molecule versus another. So let's think about, for example, methane here in this image. You can see that this is a balanced molecule. There's no difference in charge in any location in this molecule. But if we take something like water, where we have 1 oxygen and then 2 hydrogens at the, down at the bottom, oxygen is going to pull more. So it has what we're going to call a dipole moment. So the oxygen is more electronegative than the hydrogens, and that charge is not balanced. So that gives us one end of a molecule that's going to be slightly negative and one end that's going to be slightly more positive. So just think polar, think north and south pole on the Earth. There's a difference from one end to another. Conceptually, that's what polar, non-polar means. And then remind me of your name? Sabrina. Like Sabrina said, water will dissolve polar molecules, but non-polar molecules like lipids will not dissolve in water. So hydrogen bonds are a special type of covalent bond that utilizes hydrogen. Water is a good example of hydrogen bonding. So these are very weak bonds, but we have so many of them that they can be really powerful. I had a little breakthrough moment a handful of years ago, taking the shower, and you know how to keep the water inside the shower, you might take your shower curtain and put a little water on it so it sticks to the wall? Those are all hydrogen bonds. And it keeps your shower from making a mess. And I just had a moment, I was like, wow, these little hydrogen bonds are keeping me from having to clean up my bathroom floor, right? So we can see here that the oxygen is more electronegative, so we have more of a negative pull towards the oxygen and then slightly more positive towards the hydrogens. Okay, we have a couple types of chemical reactions we're going to talk about today. You can think of them simply as condensation or hydrolysis. And we're going to see a condensation reaction come up later in the chapter. So take a look here. If we take maltose and water, combine those, so these we're going to call our, what's the name of these two? Generally speaking, in a chemical reaction.
Is it the reactants?
Yes, so here on the left of our arrow, we have our reactants. They will form products. So water and maltose react to form 2 glucose molecules. And so here, what we have done is we have removed water from its liquid form and imparted it to create 2 glucose molecules. So we're going to call this a dehydration synthesis. We have, or wait, pardon me, got that backwards. This is hydrolysis at the top. Maltose and water, condensation. So I just noticed a synthesis. Pardon me, you all. I've got my textbook open, and it looks like there's a dehydration or condensation. I'll go through and I'll update these slides for you. So, hydrolysis, we're going to take water and break it to create 2 molecules.
Again.
Okay, thank you. Share, PowerPoint. There we go. And back. All right.
Thank you.
So this is, yeah, you're welcome. This is where we're spending the time on. So hydrolysis, we are breaking water, as opposed to condensation, where we are resulting in water. We're going to end up with some water that's created. If we take 2 glucose molecules, put them together, we end up with some water that results, so therefore it has condensed. And again, I'll update these slides for you all and repost it after class today. So all living organisms, as we know it, depend on water. There are some organisms, like you'll have seen those little water bears. The, what are they called? It's not actually a little water bear. They can go to space. Brine shrimp, space or sea monkeys, for example, they can exist in the Dead Sea in this really high salt content and essentially be dried out. But then they require water to be alive again, to come back to life. But some things can survive in a desiccated state, which is really fascinating. So within our bodies, all of these cellular reactions, so anything inside of a cell is going to take place in water. We, as well as bacteria, are about 70% water by mass. So water is polar. It makes for a great solvent. And so here we can see sodium chloride or table salt. These can break down into their ions, Na+ and Cl-, and then those can dissolve in water because water is polar. But if you all were to take some, say, cooking oil and pour that into a glass of water, that is nonpolar, and they will not dissolve into one another. They'll separate. Okay, we have pH scale. This goes from 0 to 14. 7 is neutral, so water is neutral out of 7. And then we have things like stomach acid that are extremely acidic, down into things like bleach, ammonia, sodium hydroxide, NaOH, extremely basic. If you hear basic or alkaline, those mean the same thing. So on a quiz, on an exam, if I say, which is the more basic molecule, which is the more alkaline, they mean the same. They are the opposite of acidic. So what an acid does by definition is it donates protons. So it's going to increase the concentration of protons in a solution. So it gives away positive charge. And a base is going to do the opposite of that. A base is going to accept available protons in a solution and decrease the amount of hydrogen ions that are available. So increase protons, you're an acid. Decrease protons, you're a base. And let's just keep in mind that this is a logarithmic scale. So as we go from 1 to 2, we actually have 10 times the amount of hydrogen atom difference. So it's a log base 10 scale. And this is just another image showing the same thing with some numbers in case you're curious. The book mentions bases, and this figure is a little bit confusing, but let's just break it down. So a buffer is a combination of a weak acid and a weak base, generally speaking. So whenever we do experiments or, in our bodies, we have chemical reactions that take place, and we might end up with some more protons as a result, or some more hydroxide ions as a result. And that, as we just saw, can change the pH. Well, our cells, our DNA, proteins are sensitive to pH. So if we have these wild fluctuations, that's going to throw off our biochemical processes. So we can have buffers that will either accept or donate protons based on the chemical reactions that are taking place. So let's say a reaction, in order to take place, needs some hydrogen ions. Part of that buffer is an acid, so it can donate those. The reaction takes place, and then after that reaction takes place, let's say that we end up with more hydrogen ions available than we need. The base in the buffer will accept those and help keep the pH relatively stable. That's not to say that it won't change, but it won't change wildly. So yeah, what you should know is that buffers are used to prevent large shifts in pH that result from chemical reactions. Okay, now getting into more of the molecular chemistry stuff. This is pretty fascinating. So of the organic compounds, there are 4 that are making up life as we know it. We're talking about bacterial cells right now. So a bacterium is primarily water, 70% water. We have a little bit of some small molecules and ions, and then about a quarter of it is all organic compounds. Of those organic compounds, more than half are proteins, followed by nucleic acids, phospholipids, and carbohydrates. Where do we have phospholipids?
In the cell wall.
Yep, so in a bacterium, we're going to call it a cell wall. In a human, we're going to call it a plasma membrane. So in that outer membrane of cells, all cells, they're going to have a double layer of phospholipids. And I don't think I have an image here, but once we get to phospholipids, RNA, right, it's the code of life. Proteins are going to...
That classroom had major internet issues. Like we...
Okay, so what I'm going to do is I'm going to go talk to our department chair as soon as class is over today, assuming she's here. If she's not, I'll shoot her an email, and I'll try and get this fixed for tomorrow, at least next week, though.
Veldon did. But
Okay.
she had talked to Dr. Doyle about the connection in that classroom.
Okay, great. Thank you for your feedback. I'll be sure to run it by her as soon as class is over today, though, okay?
Sure.
All right, here we go. Back in it. At least now I'm able to see you whenever you can't see it. Okay, so we have phospholipids, nucleic acids, proteins, carbohydrates. Carbohydrates are sugars. All right, within organic chemistry, we have parts of molecules that will branch off, and we can call those functional groups. So they're going to allow for a certain shape to take place. Shape is going to allow function. So we call it a functional group. For example, we have hydroxyl groups. So OH, if we attach a proton to a hydroxyl group, we are left with a water molecule. So we will see hydroxyl groups. We will see amino groups. Amino groups are a part of an amino acid, as well as other molecules. And we're also going to see phosphate groups. But I doubt we'll talk about carbonyl or sulfhydryl in this course. And so here we're mentioning an enzyme. An enzyme is a special type of protein, but all enzymes are proteins. Not all proteins are enzymes, but enzymes are proteins that catalyze chemical reactions. And they're going to do so by acting on the functional groups. This slide looks a whole lot more complicated than it is. This is just breaking down sugars.